Hello, good afternoon, y'all. My name is Joshua Lassero, president of ACMW, and welcome to the Day in the Life of a Bioinformatician with Veronica from Stanford School of Medicine, hosted by ACMW. You might be wondering, what exactly is bioinformatics and why is it important? Today, we hope to answer that question, but before we get started, I would like to talk a little bit more about what ACMW is. ACMW stands for the Association for Computing and Machinery Women's Chapter. This chapter was created in an effort to point a spotlight on one of the gender minority groups. However, we recognize that our name is not a requirement for entry, but as a focus of empowerment from where the creation of this chapter stemmed from. We are an all-inclusive group open towards women, marginalized genders, and minority students. However, you do not have to fit that description to join our sub-organization. ACMW is an amazing group to join if you're wanting to make new friends, just starting to get into technology, or are trying to understand the possible outcomes with your career in tech. At ACMW, we want to provide an inclusive environment along with opportunities for anyone and everyone who's interested in learning more about computer sciences. However, I understand that many of y'all tuning in are in the field of biology, statistics, environmental science, engineering, or other majors. To join our organization, you do not have to be a computer science major. And if it's your first time tuning in, we hope you learn a little bit more about computer science and how it can impact your field. No matter your knowledge in computer science or your background, we welcome all majors and everyone and anyone who's interested in learning more. Now, let's meet our team. Our team is composed of the Vice President Izzy, Public Relations Officer Mina, Secretary Natalie, Social Media Director Elizabeth, and Junior Officers Rachel and Samantha. This lovely and amazing team has been working hard with me all throughout the summer and this semester to bring events for all of y'all. And if you would like to get to know them, feel free to DM the chat. But before we continue, I will ask y'all to make sure to sign in. If you're new to ACM and you haven't signed up to become a member, please scan the ACM new member registration code. By registering, you will be eligible for swag and giveaways. And in addition, sponsors will be able to look at your resume, and that could potentially land you an internship in your field. By attending 20% of our fall meetings, you will be eligible for any of the mentioned benefits. Speaking about benefits, to those who have signed up to become a member, scan the ACMW meeting sign-in. One lucky audience member will receive a $10 gift card of their choice. To find out who the winner will be, stay tuned on our ACMW Instagram page. The winner will be announced today. As y'all scan those QR codes, I will be going over some of our upcoming events. On the 23rd of September, we will have 4 wonderful students and 1 PhD professor come and talk about their experiences in computer science in our Women in CS panel. If you're interested in computer science, want to learn more about the field, and help support your community at UTSA, I highly encourage y'all to attend. On October 7th, bring your game on. We want to meet new friends and make everlasting connections. Do you want to play board games and de-stress from school? Join our in-person event to relax and have fun. We really want to get to know y'all. On the 21st of October, we will have our sponsor, HEB, come and talk about software engineering, UI/UX design, and cybersecurity. Last but not least, save your calendars for October 23rd. Code Quantum is San Antonio's first 24 hackathon geared towards marginalized genders to promote inclusivity and diversity. Have you never programmed and want to learn how to code? Or are you experienced and want to show your skills? No matter your knowledge in computer science, join this 24-hour competition open to marginalized genders. One of our officers will be dropping the link shortly, and there will be lots of prizes, swag, and word on the street is that Google is one of our sponsors. So we hope to see you there. For more information on the following events, please keep a lookout on our social media page and Discord. Our Discord link will be sent in the chat. But now, without further ado, let's give a warm welcome to PhD student Veronica, who will be walking us through her day in the life as a bioinformatician.
All right. Hi, everyone. First, thanks for inviting me to be a speaker. I'm really excited to be here. And I'm going to go ahead and share a few slides with you. All right, so my name is Veronica Behrens, and as Joshua said, I'm a PhD student at Stanford. But of course, I didn't start out as a PhD student at Stanford, so I want to just give you a little bit of background about who I am, where I came from, and that can put some of the things I'm going to say in context. So I am from a small town in Sycamore, Illinois. It's a rural farm town. And my undergrad degree is from Harvard. I graduated in 2015, so not that long ago to me, but I might seem old to you all. And you might notice that my degree was in archaeology, so nothing related to computer science or biology, really. And this is me on an archaeological dig. You can see that there's a skeleton here, and this one is me in the bucket hat. And really, my passion for what I do now actually did stem from what I did in archaeology. So I've always been interested in what makes us human. And so as an undergraduate, I studied what makes us human by studying bones and the fossil record. And I actually decided to make a switch to study what makes us human by studying the code of life, which is DNA. And so when I decided I wanted to study what makes us human by studying DNA instead, I decided I should get some sort of laboratory experience. So after I graduated from my undergrad, I became a lab technician at Northwestern. And what I did was actually work in global health. So I was trying to develop a diagnostic tool for diagnosing tuberculosis. And this tool would be deployed in underfunded or under-resourced countries. So a very cheap way to diagnose a very devastating disease. And it was an intersection between engineering and biology. And it was a really interesting introduction to sort of traditional biology to me. Then after that, I got a master's degree in human genetics. And this is where I actually learned to code. So I did not learn to code until after I graduated from undergrad, after working for a year, and really was out of necessity. And that might sound funny to you. You're like, oh, genetics, why would it be necessary to learn how to code? And I will hopefully make that clear in a few slides. And so now I am earning my PhD at Stanford. I am in my soon to be my 5th year as a PhD student, and generally PhDs last around 5 or 6 years. So I'm a pretty advanced in my degree candidacy, and hopefully I will be graduating in about a year and a half. So now I want to just describe a little bit about why I'm here today, because what I told you is not really sounding super relevant to computer scientists, right? Well, now I consider myself sort of not a jack of all trades, but a jack of many trades. And so what I do on a daily basis integrates biology with statistics and computer science. And if you intersect these things, that is really what the field of bioinformatics is. And I want to highlight the fact that really this is the Venn diagram that data scientists will show you. So intersecting some sort of specific knowledge of some sort of specific field with statistics and computer science, and then that is data science. So really, bioinformatics is data science. It's just that the specific domain that you have knowledge in happens to be biology. And this could be, this knowledge domain could be economics or psychology or really anything to be a data scientist. But specifically, I have quite a bit of specific domain knowledge in biology. But the point of me showing you this is that even if you aren't particularly interested in biology, this sort of type of work is something that can be open to you as a data scientist and maybe currently a computer scientist, and potentially statistician too. I don't know what all of your backgrounds are. So I actually want to do a little icebreaker now to see what our audience is really like. And I want you to put in the chat, in the comments, on a scale of 1 to 10, how well did you like biology in high school? So, or now, either way. So 1 would be, I really hated it. I didn't like it at all. And 10 is like, it was my favorite subject. I want to do it for my entire life. Yeah, so I'm seeing some interesting things. So, oh, I see a lot came in. All right, so we have some who loved it, some who really hated it. Okay, we have a really broad range here. So that's really cool. Just to share with you, when I was in high school, I would say my love of biology was at about a 3. I actually really didn't like it. I felt like biology, at least in my high school, was memorizing a lot of facts that didn't seem particularly intellectually stimulating, maybe. It wasn't about solving problems or puzzles. It was just read the textbook and, learn this really complicated process that happens in your body, or memorize all these facts about the organ systems, or put a leaf under a microscope and draw it, or dissect a frog and memorize all of the muscles or something. So it really was not particularly puzzle solving or problem solving in high school. And I want to emphasize that really, when you get to college and beyond, biology really is solving problems. And that is also what coding is, in my opinion, which is solving problems using code. And so I'm going to go ahead and reshare my screen. And here it is. So what do you all think this is? I can't see the chat anymore, but go ahead and put in the chat what you think this might be. So I'm going to go ahead and assume that somebody probably put DNA. And yes, this is a whole mess of letters, and each letter represents a nucleotide, which is the fancy molecule that actually makes up all of the DNA that is found in the cells of all living things. And the human genome, and genome is just a fancy word for all of the letters that make up your DNA. So all of the DNA that is in a single human cell is made up of 3 billion of these letters. So you can imagine we have a single PowerPoint slide right here. You would actually need 3 million of these PowerPoint slides that looked like this to display the DNA that's in a single human cell. So you can imagine that if you didn't have computers to go and coding to go through and analyze this type of data, you would be lost. You couldn't do this by hand. And imagine instead that instead of just having, the 3 billion letters that make up the DNA in a single human cell, imagine that you actually have this data from, say, 1,000 patients or 2,000 patients who all have similar symptoms. And you're trying to figure out what is in common in all of the letters that make up their DNA in order to figure out what might be causing their particular symptoms or their particular disease. So we just multiplied that problem by, 1,000 or however many patients that there are. And so you can see that really this type of data is sort of a quintessential big data problem. And when you have a big data problem, you turn to computer science and statistics. And I just want to point out that this problem is actually extremely recent. So we actually weren't even able to get this whole mess of 3 billion letters per human cell. It was impossible to even get the sequence of all of the DNA in a human until 2003. And in 2003, it cost, I don't actually know how much, but like $1 million probably in order to sequence a single human. This has actually, the cost has been declining rapidly. Now it costs actually under $1,000, which when you talk about biological research is not that much, but around $1,000 to get this data now for a single person. And so it's just a very recent problem. And that means that actually there aren't very many people in the grand scheme of things, who are doing this type of work, which means that people who can integrate biology and statistics and computer science are extremely in demand, and people who are good at it are still quite rare. So, and I also want to highlight that this field is getting even more exciting. Maybe you all have heard about CRISPR. So in 2012, it became possible not just to get the sequence and look at the sequence of all of the DNA in a human, but actually to edit and change and engineer the DNA. Now, there's a ton of weird regulatory things surrounding this. There's nothing actually, no CRISPR treatments or anything that are approved in humans. But the point of me bringing this up is that this field is it's so exciting right now. I think it's the most exciting field. Of course, I'm a bit biased, but yeah, I think that this is really just going to be, with the sequencing revolution and now the CRISPR revolution, this is a super relevant and really exciting field to be in. Okay, so now what do I do then? So if you have a bunch of these letters, you know, these nucleotides, all this DNA sequence, what do I consider myself? Like, what do I do on a daily basis? Well, in the grand scheme, maybe this will sound a little corny, but I actually think it's quite beautiful. What I consider myself doing is decoding the code of life. So DNA is kind of like the code, the computer code that controls a human in a way. And so I decode that code with computer code. And the thing to know about DNA, the code of life, is that it is not a very logical code. In fact, we don't know what most of these 3 billion letters are doing in our cells. We don't know what their role is, what their purpose is. We know so little about all of the sort of mess of letters that make up our DNA. It's quite yeah, and I guess the reason is because this is such a recent field that we were only able to get this data starting in 2003. And so there's so much to decode. And of course, the code of life DNA is extremely poorly written. So with, you know, the point of making a computer like a coding language is that it's logical, it's easy to use, and that is not the case with the code of life DNA. It is not easy to tell what's going on, and it is extremely inefficient. There are things that were just carried along in your DNA that have actually no known purpose at all. There are detrimental things that happen, you know, I guess bugs in your DNA that cause really, really terrible things to happen, namely disease. And so figuring out what is actually sort of good, the parts of your DNA that are, you know, making things function normally, and then teasing that apart from the things that are making your are sort of bugs in this code of life in your DNA is a really difficult problem, and one that requires, you know, coding, computer coding. So now, what do I sort of do on a daily basis? Well, the first step of anything is to generate data or potentially find existing data. So this is the sort of thing for generating it that you might think of when you think of traditional biology. So you would think of transferring small amounts of liquid from tube to tube using a pipette. Things that you might have done in a lab course or something, like a biology lab course. It's also possible to do really cool things like grow cells in the bottom of dishes. I do that a whole lot. We also work with different animals. So maybe we have made a change to the DNA of a mouse, and now we need to, you know, collect all of the data about that mouse that we made a particular change in. So data generation is something that is pretty much in the realm of solidly biology. Of course, if you don't want to generate data, there's so much existing data out there that is just waiting to be analyzed, so you don't have to generate it yourself all the time. Okay, then the next step is to analyze it. And so this is oftentimes where the sort of statistics and computer science knowledge comes in. So you might have, for example, say that you made we'll go back to the mouse example. So say that there was we have an idea that there's a particular genetic change that is causing some sort of disease, but we don't know why it's causing the disease. We don't know really anything about it except that it's a common mutation or a common change that occurs in a lot of patients who have a particular disease. So we went ahead and used a mouse to model that disease, make that change in a mouse. Okay, so we have all of this data. How do we go about analyzing it? Well, we first might look and see if there are existing tools to analyze the type of data that we have. And if there are existing tools, then we'll use them. But another thing that a lot of bioinformaticians will do is actually develop their own tools. And so there's a lot of new type of data that biology will generate. And there is a huge amount of existing tools, but there's also some problems that can't be solved using the existing tools. So you can also do some sort of software development to analyze certain types of data. And of course, if you do a good job, you can then share that software with others in the scientific community so that they don't have to sort of reinvent the wheel every time. And this is actually how tools get shared between biologists. Someone will solve a problem, they'll publish it, make it available to the rest of the community, and then the rest of the community can actually evaluate whether they think it's a good tool or not and decide whether they want to use it. Okay, and then a big part of actually doing bioinformatics is visualizing data. So after you've analyzed it using a bunch of existing tools to plot it in creative ways, and there are some types of data that are actually really difficult to figure out how to visualize. So you can actually do things like develop web apps or command line tools and things that can actually just be about visualizing your data. And of course, you can share those with others as well. And then after analyzing and visualizing, you get back to needing some of your domain-specific knowledge. So interpreting your data. So figuring out exactly what's going on. And then finally, how does that result fit into the sort of bigger picture? And that also often requires of this domain-specific knowledge. And I just want to go back to the point that this set of steps is very general. It doesn't have to be applied to biological data. It can be applied to any type of data if you're interested in being a data scientist, which would leverage your knowledge of computer science in mainly the analyzing and visualizing parts of the data. And then you could choose what sort of domain-specific knowledge you would be interested in for generating and interpreting and, figuring out how it fits in. So I actually want to pause and ask for questions. So, I have one. Okay, so is the purpose of deciphering the code to be able to edit it and prevent diseases? Yeah, so there are a lot of ways that deciphering the code can be interesting. So one of the ways, or one of the reasons that it's interesting is to sort of figure out what is going wrong in, your DNA or the code of life, so that you can develop treatments or drugs or something to actually treat the disease. That is completely a reason to look at the code of life or DNA. But another reason is just sort of out of curiosity, and that's what drives me mostly. So I just think that learning what makes us tick which is our DNA, in my opinion, is it's just a really cool thing to do. And so I actually study how our DNA is different from other animals' DNA to figure out what makes us human and not like our most recent or most closely related ancestor, which is chimpanzees. So what of the 3 billion letters that make up our DNA, which of them caused all of the differences that we see between humans and chimpanzees? So that's, a completely different reason to study the code of life. Now, another one is related to, so I'll just bring up a completely another way to study this kind of thing. So, of course, COVID is impacting our lives, obviously, in a very extreme way. And COVID has its own genome, it has its own DNA. And so it's been the role of bioinformaticians to actually figure out how COVID has been mutating. So taking all of the patient data and figuring out which letters or which parts of the DNA of COVID have been changing and what those changes might be doing. So that's related to sort of variants. It's also the job of bioinformaticians to just sort of figure out how things are spreading. So figuring out which variants are most closely related to others to kind of figure out how COVID has been spreading. So anyway, all of that, those are all reasons to want to study the code of life. There's so many others, but okay, we have another question. So on a day-to-day basis, how many hours do I spend programming or analyzing data? Yeah, so I would say that really depends on the day. So it depends on which stage in that sort of flow chart I'm in. If I'm in the generating data phase, I might not spend any time coding that day. But if I'm in the analyzing data and visualizing data type of phase, then I might spend months. I might spend months just coding. So that's what I've been doing recently. So yeah, that's the answer. It depends. Now, I want to say that it doesn't depend for everyone. I am a very half and half type of person where I do both. You do not have to do both. You can choose to only analyze the data and develop tools that other people can use. There are so many jobs and so many people looking just for the person who can deal with this big data problem. Okay, so what programming language do I mostly use? So that's a really good question. So I mostly use Python, and the reason is I am not developing the tools for other people to use. I am using tools that other people have developed and maybe adding some sort of custom steps to the existing tools. Now, a lot of these existing tools are command line based. And so that would require, me doing some sort of bash scripting to use these command line based tools. If you are interested in being the person who develops the tools, which is a job for people who are extremely serious computer scientists like you all, those are oftentimes written in like C or C++, so like a faster, better programming language for that sort of purpose of processing a lot of data quickly. So I will also just say that R is an extremely common tool to use in bioinformatics. I know it's not exactly a real programming language, but it is extremely good for statistics. So Yes, and for visualization. It's extremely good for making good plots. And there are also a lot of packages that people in the scientific community have already developed. And so you don't have to reinvent the wheel every time if you can find a package in R or in Python that will, or a command line sort of Unix-based package that will do the job for you. So that's a really big challenge to make sure you're not reinventing the wheel every time. All right, so let's see. Another question. Coming from a non-CS background, how hard was it for me to learn computer science? Yeah, so I tried to take a computer science class when I was an undergraduate. I lasted for about 2 weeks, and then I dropped it and didn't try again until I was forced to. So I was definitely not a natural to coding. And so I guess this message is if you found it really difficult at first or are currently finding it really difficult, don't give up. I also was trying to learn C when I first tried, and I think Python is a much more friendly language to learn with, and that's the one that I ended up actually learning to code in when I was in my master's. And the reason that I actually learned to code was, like I mentioned, out of necessity. And it's because I had a project that required me to analyze all of these A's, G's, C's, and T's, all the letters that make up our DNA. And if I didn't learn, I wasn't going to be able to analyze the data, and I wasn't going to be able to make progress. So yeah. Once I was a master's student, I actually didn't find it particularly hard. I don't know what the difference was, but maybe it was the language itself, or it could have also been because I was very motivated to solve my biological problem. So, all right. Can bioinformatics provide a way to see the effects of modifying DNA of humans? Yeah. So that's a good question. So it is extremely hard. So the question is, can you use just like bioinformatics to see what changes what potentially editing the genome, what effects that would have without actually editing it? The answer is kind of no, at least not right now. And the reason is because we don't know what most of our DNA is actually doing. But something that you can do is actually make the changes. So a very common lab animal to use is mice. And the reason is because they're easy to manipulate and they're mammals. And there's actually a ton of similarities in terms of their genes and their DNA to humans. So you could make the intended edits with CRISPR in mice and then analyze the data using bioinformatics. So whenever you make a change to DNA, a very common thing to do is to then see how that affects which genes are turned on and which ones are turned off. And whenever you have that sort of data, you end up with a bunch of letters. Instead of the letters representing your DNA, they're actually representing your RNA. And so anyway, bioinformatics would definitely be involved in figuring out what CRISPR and what genetic engineering is doing, but I don't think, at least right now, you could make a bunch of predictions without actually doing the experiments. All right, so now we have some questions about AI and machine learning. So yeah, that's a really good question. It is playing a huge role recently. So there are so many data sets where if you just use the traditional methods of, analyzing your data and visualizing it, you're not going to pick up on what is actually driving the effect. And so using machine learning is an extremely good way to sort of analyze big data sets where you don't know what's going on and to find what is going on. So another way that machine learning is really taking off is for sort of automating the really time-consuming and annoying things that you're required to do if you, don't know how to code. So, for example, if there is a particular image, you have the same image. So, say for example, there's something that is making your cells, like your body is made of cells, and you can actually take pictures of the cells. So say there's something that's changing the shape of them, and you need to go through and like count the number of cells that have this different shape. Well, you can imagine a human going through and counting the cells that have some sort of weird shape, but that would be it would just be terrible if you had to do that, thousands and thousands of times. So that can be a really, really great way to have machine learning do the job for you. Another thing can be if you are interested in, I was going to give another sort of, anyway, so the answer is there are a lot of ways that machine learning is playing a role in biology. Okay, so what are some courses or experience that helped pique my curiosity? Yeah, so I would say that I took a class during my junior year that really piqued my curiosity in this, and it was just a straight genetics course. And so that really is what motivated me to make the switch from studying humans and what makes people human, instead of studying it with bones, to studying that using biological information. And so I would say that if you have any inkling in being a data scientist, whether or not it's biology or economics or something, it would probably be a good idea to see if you were interested in some other subfield. So if it's biology, take some biology courses. Another sort of, I guess I'll leave it at that. So in the future, what would I like to do with my PhD? Yeah, so that's a really good question. What careers can you have as a bioinformatician? Yeah, so what do I want to do? I'll start with that. So I personally would like to be a professor at a small liberal arts school and focus on teaching and research, but a lot of teaching. I think it's really important to pass knowledge on to the next generation, and so that's something that I would like to do. But of course, that is a very traditional thing to do with a PhD. It's not what you have to do with a PhD. So there are, as I mentioned, there are so many people looking for people who have knowledge in biology, statistics, and computer science, and are capable of analyzing this type of data. People who are generally interested in computer science, in biology, are usually not by default interested in computer science. And so finding that person who is has enough knowledge of biology, but is really interested in answering biological questions using coding, it's still pretty rare to find a person who's really good at that. And so there are so many companies just in industry who are looking for people who can analyze this type of data. And that can be private companies, it can be hospitals, it can be just really any sort of company that would require analyzing a lot of data that is also even tangentially related to biology would be interested in somebody who has this kind of skill set. You also don't have to have a PhD to try this out. So I know quite a lot of people who were pretty good coders and just decided that out of undergrad they wanted to, maybe in undergrad get some sort of internship to try this out, or after undergrad having their first job be as a introductory bioinformatician, kind of how I was a lab technician. Some people who are actually good at coding after they graduate from undergrad will try it out. Oh, thanks. I'm glad to hear that there's somebody who's taking bioinformatics. That's awesome. So yeah, I guess how can you start learning bioinformatics? So I would say that there are a lot of different paths you can take. If you already know some coding, I would suggest trying to take a class that is specific to coding and specific to bioinformatics, so that you can sort of learn how to apply what you already know to some of these sort of big data sets. Because I don't think what you learn in a straight computer science course is necessarily the same type of skills or the same type of coding that you might always do as a bioinformatician. It's just it's just a little bit of a different, you're not developing a product really, or you can, I guess you can, but you're not always just developing a product. You're trying to answer some sort of question and use the data that you have in order to interpret it in some way. So yeah, that's what I would suggest if you have some sort of background in coding. If you don't, but you have an interest in biology, then a good place to start can sometimes even be with online courses that are geared at biologists who are interested in becoming computational, because there are a ton of biologists who are interested in becoming computational. And so there's a lot of sort of courses out there that are just free, Coursera or whatever, that you could try. Or if there's one at college, that is at ACM that is geared towards, sorry, not ACM, but is geared towards towards biologists trying to become computational, potentially an R course or a Python course. That could be a really good thing to do. Okay, so, okay, awesome. So yeah, I guess my short answer would be take that class that is in the CS department. Yeah, I don't have anything else to say, but if anybody wants to ask any more questions, I can stick around. Oh, okay, I guess we have one. Have we ever edited genes in humans or only mammals? Yeah, so some people have edited genes in humans. It was actually quite a controversy. There was a Chinese scientist who sort of went against all of the international regulations and went ahead and did some editing of embryos to try and prevent the kids from getting AIDS. So it was quite controversial, and doing that sort of editing is not sanctioned by any country, including China. So yeah, he actually went to jail for a little while. But that said, that type of editing is very controversial, but there are definitely treatments that do not include modifying sort of an embryo that are, or a baby that are going to be allowed in clinical trials. And so that kind is related to curing diseases where you can actually take like blood. So one example is for sickle cell. Sickle cell disease is a disease that basically makes your red blood cells not able to carry oxygen to your body as well as they should be. And so the blood cells have a really weird shape that makes it hard to carry oxygen. And so what you can do is actually take out some like blood from a person who has sickle cell, do some sort of edit the gene that is causing this bad shape, and then inject those cells that have been sort of fixed back into the person. And this is currently, like I said, in clinical trials, and it doesn't have these sort of long-lasting effects that, editing a baby has. It's on a consenting adult, and yeah. So anyway, the answer is yes. We have edited humans. Some of the editing that has been done on humans is really great, and some is not so great. And there are a ton of ethical issues around editing humans related to, should we enhance traits that humans have? Should we be able to make people have bigger, stronger muscles, or run faster, or be taller? We actually can't make people taller yet. We don't know actually the genetic basis of height. It's there's a ton of different regions in the genome that cause height, or make someone's eyes a different color. So is that okay? Or is it only okay to, use editing to cure debilitating diseases? So it's an ongoing question. All right, so let's see. How do they know if it worked? What did he... Oh, yeah, so they don't actually know if it worked. So they know that the change was made, and to the gene that is known to cause, I guess. So the gene that they changed, they successfully changed the gene, but I guess they don't know what the potential side effects could be. So actually that gene is also protective against malaria. So there are a lot of African people who have the undesired mutation in this gene that can make people more susceptible to AIDS. Anyway, so the change in that gene also, even though it would make you more susceptible to HIV, it makes you less susceptible to malaria. So there are a lot of people in Africa where malaria is super prevalent that actually have this this change or this mutation in the gene, and it's actually beneficial for them. So anyway, it's a bit controversial. But the way that they know that it worked, I guess, is through sequencing the genome, and analyzing the DNA that the babies have. So I guess other people have heard about this controversy of using CRISPR and editing and stuff. But really, really interesting field. I also want to say that like CRISPR in general can be used for lots of interesting in just like studies. So that maybe will sound silly to some of you. So one of the ones that I've been working on is actually trying to figure out what So I study evolution, not just in humans, but in other animals, to try and figure out how our genome and our DNA has evolved, not just us, but other animals. How your DNA changes, or how DNA changes as animals evolve. Like, what parts change and what do those changes do. And so if you have a prediction about, oh, maybe this particular region of your DNA is responsible for doing, X, XYZ or whatever, you can actually make that change using CRISPR and see if your prediction is correct. And of course, you don't make that change in a human, you would make it in some other animal. So I've been making some CRISPR edits in lizards to see if a particular region is causing their limbs to be shorter, because that is advantageous for certain populations of lizards that moved to islands. So anyway, and of course, analyzing that data requires coding, and statistics. Anyway, but yeah, I also think that evolution is awesome. Can people use CRISPR unethically? Yes, definitely people could use CRISPR unethically. So yeah, and also what is classified as unethical is really specific to the person too, right? So yeah, I guess people are really concerned that I don't know if you've heard of the term eugenics, but eugenics is basically just trying to get a sort of genetically perfect person and get rid of all the people who are not genetically perfect, right? And so there's this really slippery slope of, if we change the genes of people, is that a good idea? Are we going to be, potentially being, trying to, I don't know, just trying to create a perfect race, right? It harks back to a lot of the things that are, happened in Germany with the Holocaust. So anyway, there's so much controversy surrounding CRISPR's use in humans for that reason. But there are also other ways to use it unethically on animals, in laboratories too. There's a lot of ethical considerations surrounding this, but yeah, I agree with Neon the Iguana about it being a perspective on what's good and bad.
Well, it seems like we don't have any more questions, so we're going to go ahead and conclude our meeting. Thank you, Veronica, for presenting today, and everyone for joining. If you haven't signed in, make sure you do so. And we hope to see everyone at our next meeting on September 23rd for our Women in CS panel. Also, stay tuned for our giveaway announcement on Insta or Discord. Thanks for coming.